Imagine a world where organ failure is no longer a death sentence. Picture a scenario where the fear of losing a loved one to organ failure is a thing of the past. This is not just a dream but a tangible future that is being crafted in laboratories around the world. Imagine a future where patients don't languish on waiting lists, hoping for a life-saving transplant. Instead, they receive timely, personalized care, tailored to their unique biological needs. The anxiety and uncertainty of waiting for a suitable donor could be replaced with the assurance of a readily available solution. This is the promise of bioprinting, a revolutionary technology poised to transform medicine as we know it. Bioprinting uses advanced 3D printing techniques to create living tissues and organs, layer by layer, with precision and accuracy. This technology is not just a leap forward, it is a giant stride towards a future where medical miracles become commonplace. Bioprinting offers the tantalizing possibility of creating fully functional organs in the lab, offering hope to millions suffering from organ failure. Imagine a world where a bioprinted heart, liver, or kidney can be custom-made to match the recipient's body, reducing the risk of rejection and complications. This could revolutionize the way we approach organ transplants, making them safer and more effective. This isn't science fiction. It's the future of medicine unfolding before our eyes. The advancements in bioprinting are not just theoretical, they are being realized in cutting-edge research facilities and medical institutions. The integration of bioprinting with other emerging technologies, such as artificial intelligence and regenerative medicine, is paving the way for unprecedented medical breakthroughs. Scientists and engineers are working tirelessly to refine this technology, inching closer to a reality where organ transplantation is no longer limited by donor availability. The collaboration between biologists, engineers, and medical professionals is driving innovation at an unprecedented pace. Each day brings us closer to a future where the limitations of traditional organ transplants are overcome by the limitless possibilities of bioprinting. The potential to alleviate human suffering is immense, marking a new era in medical innovation. The impact of bioprinting extends beyond organ transplants. It holds promise for regenerative medicine, personalized treatments, and even the development of new drugs. As we stand on the brink of this new era, the hope and excitement are palpable. The future of medicine is here, and it is brighter than ever before. At its core, bioprinting is like building a house, but instead of bricks and mortar, we use cells and biomaterials. This revolutionary technology is transforming the field of regenerative medicine, offering new hope for patients in need of organ transplants. Just as a house provides shelter and structure, organs are essential for the proper functioning of our bodies. The process of bioprinting is intricate and requires a deep understanding of both biology and engineering. It's a meticulously orchestrated process that begins with creating a blueprint, a digital three-dimensional model of the organ we want to create. This model is designed using advanced software that allows scientists to map out every detail, from the overall shape to the intricate network of blood vessels. The blueprint serves as a guide for the bioprinter, ensuring that each layer of cells is placed with precision. This blueprint guides the bioprinter, a sophisticated machine that acts like a robotic arm, carefully placing cells layer upon layer. The bioprinter operates with incredible accuracy, depositing cells in a specific pattern to mimic the natural structure of the organ. Each layer is built upon the previous one, gradually forming a complex, three-dimensional tissue. Imagine a cake decorator meticulously piping intricate designs. Just as the decorator must have a steady hand and an eye for detail, the bioprinter must operate with precision to ensure the cells are placed correctly. The process requires a delicate balance of speed and accuracy, as the cells must remain viable throughout the printing process. Similarly, the bioprinter deposits bio-ink, a special mixture containing living cells, onto a scaffold. This bio-ink is carefully formulated to provide the necessary nutrients and support for the cells to thrive. The scaffold acts as a temporary mold, providing structural support as the cells begin to fuse together. Over time, the scaffold will degrade, leaving behind a fully formed organ. This scaffold acts like a temporary mold, providing structural support as the cells fuse together, gradually taking the shape of the desired organ. The process of cell fusion is critical, as it ensures that the cells form a cohesive and functional tissue. Scientists must carefully monitor the bioprinting process to ensure that the cells are behaving as expected, and that the organ is developing correctly. It's a delicate dance of biology and engineering, a testament to human ingenuity. The collaboration between biologists and engineers is essential, as each brings a unique set of skills and knowledge to the table. Together they are pushing the boundaries of what is possible, creating organs that could one day save countless lives. The future of bioprinting holds immense potential, and we are only just beginning to scratch the surface of what this technology can achieve. The implications of bioprinting for medicine are profound, promising to revolutionize healthcare in countless ways. Imagine a world where patients with failing kidneys no longer have to endure years of dialysis, their lives tethered to a machine. Bioprinting offers the hope of creating personalized organs, tailored to each patient's unique needs, minimizing the risk of rejection. Beyond transplantation, bioprinted tissues and organs could accelerate drug discovery and development. Imagine testing new medications on bioprinted human organs, eliminating the ethical concerns and limitations of animal testing. This could lead to safer, more effective treatments, bringing us closer to cures for currently incurable diseases. Section 4. Bioprinting in Action, From Skin to Hearts. In this section, we delve into the fascinating world of bioprinting, a revolutionary technology that is transforming the field of medicine. From creating simple tissues to the ambitious goal of printing entire organs, bioprinting is pushing the boundaries of what is possible in healthcare. While still in its early stages, bioprinting has already achieved remarkable milestones, hinting at its enormous potential. The process involves using bioinks made from living cells to create structures that mimic natural tissues. This technology has the potential to revolutionize organ transplantation, reduce the need for donor organs, and provide new solutions for patients with chronic diseases. Scientists have successfully bioprinted skin grafts for burn victims, offering a less painful and more aesthetically pleasing alternative to traditional skin grafts. These bioprinted skin grafts are created by layering bioinks in a precise manner to form a structure that closely resembles natural skin. This not only reduces the risk of rejection, but also promotes better integration with the patient's own skin. These bioprinted grafts integrate seamlessly with the patient's own skin, promoting faster healing and reducing scarring. The ability to customize the grafts to match the patient's skin tone and texture further enhances the aesthetic outcomes. This advancement is particularly significant for burn victims, who often face long and painful recovery processes with traditional grafts. The holy grail of bioprinting, however, remains the creation of complex solid organs like hearts and livers. These organs are composed of multiple cell types and intricate structures, making them challenging to replicate. Despite these challenges, researchers are optimistic about the future of bioprinting and are working tirelessly to overcome the technical hurdles. While significant challenges remain, researchers have made encouraging progress, bioprinting miniature versions of these organs. These mini-organs, or organoids, are used to study the development and function of human tissues in a controlled environment. They provide valuable insights into how diseases develop and progress, and how different treatments can affect these processes. These organoids hold immense promise for drug testing and disease modeling, paving the way for personalized medicine. By using bioprinted organoids, researchers can test the effects of new drugs on human tissues, without the need for animal testing. This not only speeds up the drug development process, but also provides more accurate results. Additionally, bioprinted organoids can be used to model specific diseases, allowing researchers to study the underlying mechanisms and develop targeted treatments. The future of bioprinting is bright, and its potential to transform medicine is truly exciting. Section 5. Ethical Crossroads, Navigating the Moral Maze of Bioprinting. As with any transformative technology, bioprinting raises profound ethical questions that society must grapple with. Who will have access to these life-saving organs? Will bioprinting exacerbate existing healthcare disparities, making advanced treatments available only to the wealthy? These are not easy questions, and they demand careful consideration. The ability to create organs in the lab also raises questions about the definition of life itself. Where do we draw the line between creating tissues for therapeutic purposes and creating life in the lab? These ethical dilemmas require open and honest dialogue involving scientists, ethicists, policymakers, and the public. Section 6. Safety and Regulation, Ensuring Responsible Innovation. Ensuring the safety and efficacy of bioprinted organs is paramount. Rigorous testing and regulatory frameworks are crucial to prevent unintended consequences and protect patient safety. The FDA and other regulatory bodies play a vital role in establishing clear guidelines for bioprinting research and clinical applications. Standardized manufacturing processes are essential to ensure the consistency and reliability of bioprinted organs. Just like pharmaceuticals, each batch of bio-ink and every bioprinted organ must meet stringent quality control standards. This will foster public trust and pave the way for wider adoption of this transformative technology.